definitely have a lemon for a computer. Okay. So need to continue on with plant defenses. And um, we've been talking, kind of zooming in on the idea of primarily herbivores, something coming along and chewing on the plant. And I think we left off here, looking at the idea of this caterpillar starting to attack the plant and the idea that it does kick off a cascade of plant defenses. And so we've talked about the idea of the jasmonic acid hormone getting produced locally, right where the caterpillar is chewing, but then that jasmonic acid can go systemic through the rest of the plant with the idea that the whole plant needs to get up and running to defend itself, because chances are, if there's one caterpillar on this leaf, there'll be more caterpillars coming. Plus, this caterpillar could move on to some of the other leaves. And so it does make sense that their whole body starts to get up and running against this particular herbivore. They're also showing the idea of electrical signaling can go systemic as well to help with getting the plant up and running for its defenses. And then I think we left off here with the idea that you have the caterpillar chewing. We talked already about how the caterpillar gives off certain chemicals in its saliva that the plant can detect. And that's one way that the plant is telling or being able to perceive that it's a caterpillar attacking them. This other idea, though, these vibrational signals, when you think about herbivores chewing on the plant, that is a vibration on the plant, and that can be picked up. Now, initially, when they were looking at this was kind of a far-fetched idea. A lot of scientists dismissed it and said, nah, no. But a group of scientists decided to follow up on this a little bit more closely, and they were tracking the production of glucosinolates, because that's a secondary metabolite made in defense. And then anthocyanins are also their pigments, but they can also be secondary metabolites that can be used for defense purposes. So that's what they did. I think they worked with Arabidopsis. And so that plant, if I know that at least one group's working with Arabidopsis, but very small, petite plant. It has kind of a rosette center. That's what the RC is. And then there's leaves that come off. And what they did is they had a recording of a caterpillar chewing, and they clamped special clamps onto the playback PBL, playback leaf, and turned on the vibration recording. And they looked to see what would happen in that leaf with that vibration happening on that leaf. And then they looked to see if any of the other leaves, so they chose a leaf across from the plant, so through the rosette center and across on the other side of the plant. And that's their so-called systemic leaf that they were also testing for production of secondary metabolites. And so the light tan or gray boxes are your none, that's your control, where they were looking at no playback of the vibration or the chewing vibration playback. And you can see where they have it asterisked. That means it's statistically significant difference between those two bar graphs. And as well, so both the playback leaf and the systemic leaf were showing that the vibration was ramping up the glucosinolate secondary metabolite. And then they were also looking at the anthocyanin. And again, what they were doing in this part of the experiment, they were saying, well, okay, so vibration, what about other vibrations? Plants experience other vibrations too, like wind blowing across their leaves, or this is a leaf hopper insect that was just sitting on the leaf doing a mating call for, it was mating season for the leaf hopper, or a chewing vibration, or nothing. And so they were looking at, can the plant, does it always just kick up secondary metabolites under any vibration, or is it specific to getting chewed on? And they were showing, again, statistically significant differences between the chewing vibration over any other vibration or over the control with no vibration in terms of the anthocyanin production. And so the wind didn't work, the mating song of a leafhopper didn't work to ramp up the anthocyanin. So it was a very nice initial study that was done, and it was actually from this paper published in 2014 by Apple and Colcroft. And so they worked together in the same lab. And so when they gave a presentation on this at a local, well, it was a national plant biology conference, this was like standing room only because this was such a novel idea that most people just, like I said, they kind of just dismissed it and said, no, this can't happen. And so this paper was well done and really met with a lot of excitement, some skepticism too. And then other scientists took this up and started thinking more broadly about it and like, okay, what does this mean? You know, how consistent is this? And so then there's been several papers published since then where they're actually calling it plant bioacoustics now, where they're saying that plants are paying attention to vibrational patterns through sound. And so if you really think about what sound is and the definition of sound, it's a vibrational pattern. And so they're saying, yes, plants can pick up on that. And they wonder now, are plants able to communicate with other organisms in this way? Acoustic communication in plants. Is it a way for them to figure out if they're getting chewed on, like that other paper? And so they're really starting to delve into this more, and they're starting to figure out the mechanism that might be going on for the plant to pick up vibrational patterns. And they actually think it's the trichomes might be a good part of the plant body where it could be nicely sensitive to vibrational energy and can be used. And so these are some more recent studies where they're getting, this one is very technical. But again, zooming in on the trichomes, looking at maybe that's how the plant is picking up vibrations. And so there's this idea that, okay, if we could expose plants to certain vibrational patterns, could that cause the plant to ramp up its secondary metabolites against an insect? So like if you're a grower and you are out walking your fields and you start to see, looks like the pest caterpillar population starting to come into my field here. And if you could get that early enough where you could prime the plant with sound instead of pesticides, spraying the pesticides on the caterpillar and dealing with all the consequences of pesticide use, could you prime the plant ahead of the infestation? You know, if the caterpillars are coming, get them primed ahead of time by using sound and or some other appropriate vibration. And so they're looking at this idea. There's been some nice studies over in Japan and Europe in general, and looking at using sound. And I think, you know, if you think about it, it's way cheaper than buying pesticides for sure. It's not nearly as damaging to the environment as pesticides can be. And they've actually found that it gives extra benefits they didn't realize at the time, in that, you know, because it's ramping up secondary metabolites, not only is the plant able to defend itself, but then the nutritional quality is better for the human that eats it or whatever animal is going to eat it. And so in general, it's the case that secondary metabolites in plants are good for our health. When we eat plants that are rich in secondary metabolites, like polyphenols, antioxidants, all of that is good for our health. So it's not only helping them to defend, but it's also creating a higher quality product that they can sell and make profit off of. And so it's definitely a win-win. They've come up with a plant acoustic frequency technology generator over in, this one's over in China. And so they're starting to play around with this and even out like in field grown crops, where they're looking at the production of sound and the impact it has on secondary metabolites and helping that plant defend themselves against a potential whatever pathogen is out there or herbivore is out there. So this is young research, but it is something that is gaining interest. Here this is a paper published, Sound Waves Affect Flavonoid Contents in Sprouts. And so again, this is a pretty simple idea, really, in terms of, just get sound waves out there and call it good. And so they were showing how the flavonoid content did increase. And so they're, even if there's not like in this particular case, they're saying we could just use this even if it's not to help the plant defend themselves. Like this is just a way to increase the functional food, the nutritional quality of the plant. So they're saying, we can use this independent of getting the plant to defend itself. So interesting ideas out there. I think you'll see more of this idea, and I think it's attractive because it's much cheaper, especially if you're trying to help the plant defend itself. It's much cheaper than the chemical sprays that you have to do. Okay, so just like with us, we have a very sophisticated immune system, and we're a highly complex multicellular organism. Plants are also, have many different tissue types. They have, they're very multicellular, of course, and especially if you think about some of these huge tree species. And so it certainly makes sense that they can have, several ways to get these systemic responses to pathogens or herbivores coming in. And so, again, it makes sense that they're going to start using some of their hormones as a way to get that signal through the whole plant body. And so usually it's produced at the site of damage, and then will start to course through usually through the phloem or the xylem. And they're starting to track electrical signals. They're slower in plants compared to humans, but they're there. They now know there's vibrational signals that the plant is paying attention to and is using as a good perception of being attacked. And then now they're starting to look at all kinds of volatiles that plants are making and excreting, especially through their stomata. And so the VOCs, volatile organic carbons, you might have heard that term. So what they're kind of zeroing in on are things that are called green leafy volatiles that the plant makes and then gets out through their stomata. A lot of them are terpenoids or phenolics, well, and alkaloids too. And so there's this class of volatile chemicals that it doesn't have to be the chemicals like jasmonic acid that's, dissolved in the fluid of the plant body. It can be volatiles that are given off. And if you think about it, that kind of would be nice if you are, for example, a giant redwood or a giant sequoia, where your canopy is just huge, where you have a huge canopy. You could imagine it might take a while for jasmonic acid to get through inside the plant. If you could send off volatiles, like if maybe your lower branch is getting attacked by some beetle, and if you could then give off a volatile that can then float up to the rest of your canopy and warn the rest of your body, hey, this is happening, you better defend. And so they're starting to think that volatiles are also heavily involved in helping that plant, especially if they're big, get the whole systemic response. And then they're looking at volatiles that go out from one plant and get perceived by a nearby plant, and how that might also allow for this signal to get sent to other plants to warn them. And initially, when that question was being asked, again, they were like, no, that doesn't happen. And then a group of scientists did a pretty nice study where they were showing that it can happen, where one plant can send off a volatile that a nearby plant can pick up on and respond appropriately to that signal. And when that got published, it met with a lot of pushback and a lot of skepticism. So then they went out and did some more experiments. Now it's pretty much given as, okay, yes, this is happening. But it took a while for the scientific community to accept it, I guess, enough data to come in. So what they're showing here, I don't know if you can see, like here's the caterpillar on the leaf right here. And what they're showing, HIPV means herbivore-induced plant volatiles, okay? And so what they're trying to show is, yep, the caterpillar's chewing away, the plant will produce some volatiles, not only in this case, we talked about this already, it's attracting the predator of the caterpillar to come along and, come eat this caterpillar so it gets off me. And so that's a very slick little mode of defending itself. And then they're showing that these volatiles are repelling other herbivores. They're showing the volatile going to a nearby plant and telling those plants to go ahead and get ready because you're probably going to be next under attack. And then down here, they're trying to show some little insect chewing away on the roots. And in this case, these little squiggly things are nematodes that actually can help attack the caterpillar. And so they're saying that not only can you have above ground volatiles going out through the stomata, but you can also have that happening in the root zone. So initially they were very much focused in on what's going out through the stomata. But then some of the other scientists said, well, let's see what's happening down in the roots. And they're also showing it there as well. So the more they look, the more they seem to find in terms of the plant's sophistication in its ability to defend itself, get its whole body up and running in defense, warn others. And there's still some debate about, is the plant getting attacked actively warning others, like that's on purpose, or is it more of it's just kind of a consequence of getting attacked and other plants happen to pick up on it? And so that's a whole nother topic, but I don't have time to get into that. But anyways, they definitely can track it, that these are signals that other plants can perceive and act on appropriately. And then this is not from your book, but it has lots of stuff on here showing, again, kind of zooming in on the volatiles, like what is going on with the volatiles. And this is encompassing more than just defense. They're also looking at it's helping to attract pollinators. And so they're kind of looking at what can volatiles that the plant-made volatiles, what do they do for the plant? And they're starting to see, I think they've always known that volatiles are given off by flowers to help pollinators come along. That's been well known, but now they're starting to branch out into all these other mechanisms of why the plant is giving off these volatiles. So this is a good kind of summary slide, getting at some of those ideas, both above ground and below ground, either to defend themselves directly about whatever's attacking them or to, call in the predator of that herbivore that's chewing on them. So lots of ideas out there that they're examining more closely. So insects, definitely insects are herbivores. They love to attack the plant. Either, there's different kinds we went through, there's precision herbivory, and then you go all the way up to the chewing insects that can cause a lot of damage. And so what I've been talking to you is mostly geared toward that. When herbivores are chewing on plants in one way or another, how is the plant defending itself? Now I'd like to turn to, okay, what if it's like a bacteria or a fungus where, they're not an animal chewing, it's a little different system of how they attack. And so they find that plants will defend themselves against bacteria and fungi and viruses, but they do it in a different manner than what we've been talking about when they are being attacked by herbivores. So here are some different ideas of how pathogens like, bacteria and fungi might be entering into the plant or causing trouble for the plant. And so here's a lovely little aphid here. And I don't know if you can see, but it's got its little mouth, its stylet mouth part that it's using to pierce. So aphids are precision herbivores. And so they're piercing into the phloem, but you have to appreciate that their mouth has microbes in it and their saliva does. And so as their mouth part interacts with the plant tissue, that can be a way of introducing different microbes into the plant. So that's definitely a strategy. And we know there's all kinds of insects that feed on plants. And so whether it's a caterpillar, they have microbes in their mouth too. And so it's not just the caterpillar chewing, perhaps there's microbes coming in as well that can complicate things for the plant. Then you're seeing a leaf here in the left picture where they're showing fungi attacking and bacteria. And then this is a potato tuber on the bottom. And all these little like darker spots are lenticels. If you've taken Plant Kingdom, you've heard that term. Lenticels are areas on the bark that are tissue that's kind of loose. It's loosely organized tissue to allow for some better airflow in those patches on the bark. And some lenticels you can see very easily when you look at a tree stem, for example. Others are difficult to see. But really, when you look at a potato, a white potato, if you've ever looked at the surface, they have lenticels too, because the skin, when we say the skin of the potato, that's actually bark tissue that you're looking at, because a tuber is really an underground stem. And so all those little bumps are lenticels that are helping the airflow. And then sometimes you see them a little bit more pronounced like this and darker, because this is young stages of potato scab disease that you're looking at. And so we tend to see potato scab disease near the lenticels because it's loosely organized tissue, so it's easier for the pathogen to get in when you have a looser organization like that. So that's kind of a prime spot for really tiny organisms like bacteria and fungi. So we have to think about that as well. Here's a blow up of that image. And so again, they're trying to show fungi. Fungi are pretty amazing. They have a lot of different funky enzymes that they can use to directly attack plant, like the waxy covering, for example, and then get right into the epidermal cells. And then you're seeing the red, coming into the leaf tissue here. And then they're trying to show the bacteria too on the surface, and that they might be able to get through. They have a little tougher time than fungi. They often need like a wound to get in, or the stomata is another good avenue for them to get in. So there's a variety of ways that these microbes can get in. And sometimes it's a good thing, because sometimes these are endophytes that are going to help the plant. But in this particular topic, we're zooming in on the bad guys, where they're going to get in and they're going to start causing problems. So of course, they have to throw more jargon at us. That's going to be a little different than what we've already covered. So when you have bacteria and fungi attacking the plant, we say that they produce effector molecules, which really, it's a type of elicitor. I've already introduced the elicitor idea. It's just a subcategory of that. But depending on the microbe, these effectors can be something like an enzyme that the fungus uses, for example, to get access to deeper into the plant tissue. Or it could be a toxin. It could be a growth regulator to mess up the growth cycle of the cells and tissues of the plant in that area, so they get some nutrition from that. But basically, in a nutshell, effector molecules are something that the pathogen has that is allowing it to successfully colonize and start to get nutrition from the plant. That's the goal of the pathogen. That's what they're trying to do. Set up camp and get access to nutrients so that they can stay alive, they can reproduce themselves. And so the effector molecules is kind of an umbrella term. They're really a type of elicitor that we've already talked about when we looked at the herbivores. More terminology, MAMPs. They talk about MAMPs, which are microbe associated molecular patterns. So basically what this means is they're saying that the plant can cue in on like bits and pieces of the fungi or the bacteria. And so things that are different from the plant. So for example, flagella. Many bacteria have flagella, and the plant can pick up on that. And I'll explain a little bit more about that. Chitin is something that fungi have as part of their cell wall. And again, the plant can pick up on that. So these are MAMPs. The plant has special receptors for their ability to pick up on this stuff. Okay, so they're called PRRs, plant recognition receptors. These are typically found on the plant surface, cell surfaces. And so they kind of stick out. And so if a bacteria flagella docks into the receptor, for example, or a piece of the flagella docks into the receptor, that is the signal that the plant pays attention to. And then it will start a signal cascade to help defend itself against that particular bacteria, for example. So it's a good idea if you're a plant that you'll have these receptors on the surface like that so that you can pretty quickly tell, uh-oh, there's something here. There's a bacteria or fungi right here in my external environment. I need to try to defend myself. And so this process where they're paying attention to, they have these receptors on the surface and they're paying attention to something coming from the bacteria or fungi, that is called MAMP triggered immunity. Okay, and I need you to know what I'm talking about with that. So when we say MAMP triggered immunity, it's this idea right here on this last bullet point that something coming off of the, typically it's something from the cell structure of the bacteria or the fungus that the plant recognition receptor interacts with. And as soon as you have that docking into the receptor protein, then the plant goes, ah, okay, I have a fungus here and it's about to attack me. I need to ramp up my MAMP triggered immunity. So I'm going to show you a slide here that looks busy, but we're just going to look at part of it. So MAMP triggered immunity is happening over here, right? So here's the MAMPs. Here's, they're trying to show some bacteria and fungi hanging out. They've gotten to the surface of the plant tissue and there's something coming off of them. Whoops. Something coming off of them that these receptors, the pattern recognition receptors. So these MAMPs will dock in there and interact with the receptor. And now that's the signal cascade, you know, danger, danger, Will Robinson. You have this signal cascade then that's going to go internal. And ultimately, you want the plant to shift its gene expression so it starts to ramp up and defend itself. So there's your MAMP triggered immunity. So that's pretty much what we were just describing on that previous slide in text. Okay, another thing that they're showing here are these DAMPs. Damage associated molecular patterns. So this is nice too, that if, let's say a fungus is attacking and it's releasing enzymes and it's starting to cause cell lysis. So the plant cells are getting damaged by these enzymes. Well, that's what they're trying to show here, that you have, you know, the fungus maybe is releasing these cell wall degrading enzymes and they're trying to show that there's damage here, these holes in the cell wall. So that's, let's say that's caused by a fungus. So now what's happening is there's bits and pieces of the plant's own body that these are damage associated with. So that's bits and pieces of its own cells that are then also going to dock into these PRR receptors. And so it's, you know, effectively when it gets bits and pieces of itself docking into these receptors, it's like, oh, I'm getting chewed on because now I'm getting broken down. There's bits and pieces of me floating around here. I'm getting attacked. All right. So that's also a really good strategy that they can pick up that they're getting attacked and they're starting to fall apart, so to speak. So there's that going on as well. So you have to imagine that if you've taken any kind of microbiology class and you've tried to look at microbes under the microscope, they're tiny. You need the oil immersion lens, you have to put oil on there. And even then you're doing your fine focus and you're searching for tiny little organisms. So you have to appreciate how these bacteria and fungi, but especially bacteria, if your stomata are open, that's not that much of a challenge for them to get in through the stomata. So they can definitely get in and start causing problems. I mean, look at that, one stomata and you see a whole population of microbes easily fitting in just to one stomata. And so this is not terribly difficult. And so it makes sense that the plant has an array of different mechanisms in place ready to go to help defend itself against these types of invasions. So sometimes, you'll see that the fungus or the bacteria, it's able to avoid the MAMP triggered immunity. It's able to co-evolve. So very often what you see happening is some of these pathogens will start to co-evolve new strategies that the plant doesn't know about yet. And so that's what they're trying to show with this arrow up here. They're saying that there's pathogens gain new effectors and virulence functions. So what they're trying to show is that there's this coevolutionary process between the plant and the fungus or bacteria where the fungus pathogen, let's say, comes up with a new strategy that catches the plant off guard. Then the plant has to figure that out and co-evolve and come up with a new strategy. And so there's this back and forth coevolutionary selection pressure going on that is constantly going on. And so what they're trying to show here is that sometimes you'll get a pathogen that co-evolves well enough that they can kind of sneak around the whole MAMP triggered immunity. I kind of think about it like if you're trying to get into a nightclub and there's a bouncer there and they're like, no, you're not on the list. And so you sneak around through the back door and someone lets you in. That's kind of what I think of with this idea here, where now on this right side of the image, they're showing this idea that the fungus, let's say, has snuck past and they're actually able to get some of their toxins or enzymes or nastiness, virulence factors to get into the cell. So now it's a little bit more successful at this stage. And so what they're trying to show here is that the plant has co-evolved and said, well, okay, I'm going to, have some R proteins floating around in my cells. So the R proteins are from the plant, and what they're doing now is they're trying to catch the effectors coming from the fungus, let's say, maybe it's a toxin of some sort or a virulence factor. And you can see they're trying to show how it's binding into the receptor. And then once that binding into the receptor happens, that is the signal to the plant, uh-oh, now we're in trouble. We need to ramp up. And so this half of the image is getting at this effector triggered immunity. So this is more the idea that the microbe, something coming from the microbe has made it into the cell. And the plant has to have additional resources. And in this case, it's the R proteins floating around in there. And when you get that docking into the R protein, something coming off of the bacteria or fungus, maybe toxin of some sort, might be, it could be a lot of different things, but that will kick off what they call effector triggered immunity. So it's more than one level of immunity that the plant has. Yes, it has this MAMP triggered immunity, which is more, picking up on things outside of itself. And that's really good because that can give you early warning, something's up. But then some pathogens can bypass that. They're successful enough. They evolve new strategies. They get part of them in, or maybe there's some virulence factor in. But now there's R proteins there that the plant can make in response, and that can help them deal with the initial success of the pathogen, but then they can catch it with this like second layer of immunity. So not quite as sophisticated as us. We have, if any of you have taken immunology, you know that we are amazing organisms and that our immune system is just astounding. So not quite that sophisticated, but this is still a quite successful strategy for the plant to have more than one level. And so this is describing what we just looked at. If those effectors coming from the fungus or the bacteria can bypass that initial, the receptors on the surface, and these effectors get in, then the R proteins inside the cell, they'll take care of it, hopefully, and will mount a effector triggered immunity defense. So I'd like you to know and think about, what's similar and different about these two, I guess, layers of immunity that the plant is able to express. And unfortunately, yes, it's jargon, and scientists love their jargon. So I think this image can help kind of do the compare and contrast of the two, the MAMP versus the effector triggered immunity. And then this I found in a different resource. I don't know if this would help, but this is basically saying what we just looked at in the previous image. It's looking at, the MAMP coming from the microbe, recognized by the cell surface, the PRR receptor, but maybe the fungus has a way to deal with that and kind of outwit and sneak by. And then inside the cell, there's some R proteins that can recognize those and hopefully suppress with effector triggered immunity. So this is saying basically the same thing as the last one, just organized a little differently. So if that organization helps you, great. But this is saying pretty much this. Okay. Okay. So we know that plants don't have all of this kind of defense responses up and running all the time. It would be way too energy intensive, resource exhausting. It would just be way too much for the plant. So we say they're induced defenses. They will only kick on when they're needed. And again, we're talking about the idea of signal transduction pathways in order for the plant to respond to the initial signals that it's getting, with some kind of elicitor or effector that it can pay attention to. And so once the plant cues in on that, that kicks off the signal transduction pathway. And what, ultimately the reason for that is so that the plant starts to shift its gene expression pattern to respond appropriately to that particular threat. And so it's not a one size fits all response. The plant can be very targeted on, okay, it's a caterpillar chewing me, so then I must do my jasmonic acid and on and on. Or, oh, no, it's a fungus attacking me. So now I'm going to do, and we're going to talk about what they can do with fungi and bacteria, but it can be very targeted in that way. And so they have to have a lot of receptors out there on their cell surface, as well as in their cells that can pick up on these signals coming from the herbivore or the microbial pathogen. And that is once you get that binding into the receptor, that's really what is the kickoff event to getting that signal transduction pathway going in the plant body. And when they look at all the genes that are necessary to synthesize all these receptor proteins that the plant is using to assess what's going on around them, it's huge. It's a lot of the percentage of DNA that's devoted to making these receptor proteins. It's a lot. It's one of the largest gene families in plants. So when you look at what is all the DNA doing for the plant, this is a major portion of the plant DNA is devoted to having these different receptors that can capture all these different signals. And I mean, you just think about all the different insects attacking the plant, lots of fungi, bacteria, viruses. It's phenomenal how much they get attacked. And so they have a huge amount of their DNA devoted to, we have to make a lot of these receptors so that we can pick up on all these different cues coming at us. Oops, that was weird. Okay, so signal transduction pathways that we talked about in a different slide set, I'm trying to show how that whole system now is kicking in when the plant is, in a very sophisticated way, being able to tell exactly what is attacking them. Okay, so this image is a little bit older, so I typed that in there. This is an older slide, but the reason I put this in here is that I wanted to show kind of the progression of the science going on where, and I like this, how the artist drew this, where they're showing an elicitor idea coming, it's like a bit or piece of the pathogen. And then they're showing the receptor is going to interact with that elicitor. And then you get your signal cascade, your signal transduction cascade. So this, you can see there's question marks here. Well, this is the newer version of that slide now that's in your book. And so you can see they've pieced it out or, put in more detail of what's going on here. And so again, they're trying to show, I like the other drawing better, but again, something from the pathogen that the plant is picking up on because of all these different receptors that they have, either in the cell membrane or in the cell. And then you can see there's a whole bunch of arrows here and lots of stuff that are kicking in response to getting attacked. So we'll get into this on Monday. We'll look at all the stuff here in the white boxes. We'll get into explaining what all that means. Okay, so, but we'll quit now. We've run out of time. So have a good weekend.